Good morning, students. Today we'll discuss some important terms used in the immunology. First important term is FCA, that is Friend's Complete Adjuvant. Friend's Complete Adjuvant is also known as CFA, that is Complete Friend's Adjuvant. It is water in oil emulsion containing killed microbacterium. Greatly enhances immune response to the immunogen. Typically, it is used for initial injections. Disadvantage of FCA are difficult to mix with the immunogen and can cause tissue necrosis at injection sites. Basically, adjuvants enhance the immune response to compounds that are already immunogenic. They do not confer immunogenicity to non-immunogenic haptens. To make prospective antigens more immunogenic, it is necessary to conjugate them to carrier protein or some other complex immunogenic molecule. So adjuvants are non-specific stimulators of the immune response. When mixed with an antigen or immunogen, adjuvants help to deposit or sequester the injected material, thereby helping to increase antibody response. Next term is IgE. Immunoglobulin E is a type of antibody that has only been found in the mammals. IgE is synthesized by the plasma cell. Monomers of IgE consist of two heavy chains, epsilon chain, and two light chain with heavy chain containing four Ig-like constant domains. IgE main function is immunity to parasites such as helminthes like Schistosomia mansoni, then Trichinella spiralis and Fasciola hepatica. IgE also has an essential role in type 1 hypersensitivity, which manifests in various allergic diseases such as allergic asthma, most type of sinusitis, allergic rhinitis, food allergy, and specific type of chronic urticaria and atopic dermatitis. IgE also plays a pivotal role in response to allergens such as anaphylactic drugs, bee stings, and antigen preparation used in desensitization immunotherapy. So in this diagram we can see that the B cell, with the B cell, the ragweed pollen is attached to the receptor. The first time allergy-prone person runs across allergens such as ragweed leads to the formation of IgE. The plasma cell forms IgE against the ragweed. Then the IgE molecule attach themselves to the mast cell. The second time that person has brush with ragweed, then what happens? On the mast cell, the IgE are present, right? The ragweed molecule, when bind with these IgE, it leads to the chemical mediators such as histamines and cytokines into the environment. And then these chemical mediators cause characteristic symptoms of the allergy. Next term is IFA, that is incomplete Friend's adjuvant. Incomplete Friend's adjuvant is also known as IFA, that is incomplete Friend's adjuvant. It is water in oil emulsion without microbacterium. It enhances immune response in the booster injections, typically used for subsequent boosts after initial injection with FCA. Its main disadvantages, they are difficult to mix with the immunogen. So again, the adjuvants, they are non-specific stimulator of the immune response. When mixed with the antigen or immunogen, adjuvants help to deposit or sequester the injected material, thereby helping to increase the antibody response. To make prospective antigen more immunogenic, it is necessary to conjugate them to carrier protein or some other complex immunogenic molecules. Next term is opsonization. Opsonization is a process by which the pathogen is marked for ingestion and eliminated by the phagocytes. C3b is a major opsonin of the complement system. Tau, C4b and C3b also have opsonizing activity. The amplification that occurs with C3 activation results in a coating of C3b on immune complexes and particulate antigen. Phagocytic cells as well as some other cells express complement receptors, that is CR1, CR3, CR4, that binds with C3b, C4b, IC3b. Antigen coated with C3b binds to cell bearing CR1. If the cell is a phagocyte, for example a neutrophil, monocyte or macrophage, phagocytosis will be enhanced. So this is a bacteria in this diagram. It is bounded with IgE. It leads to the activation of complement. Then C3b, they will bind on the surface of this bacteria. And on the phagocyte we have got a receptor known as CR1. CR1 binds with C3b and ultimately it is responsible for ingestion of this bacteria with the formation of phagosome into the phagocyte, leads to the digestion. Next terminology is CD4 cells. T helper cells, they are also known as CD4 cells. They play a very important role in the immune system, particularly in the adaptive immune system. They help the activity of other immune cells by releasing T cell cytokines. These cells help suppress or regulate immune responses. They are essential in B cell antibody class switching, in the activation and growth of cytotoxic T cells, and in maximizing bactericidal activity of phagocytes such as the macrophages. So mature T helper cells express the surface protein CD4 and refers to as CD4 plus T cells. Such CD4 plus T cells are generally treated as having a predefined role as helper T cell within the immune system. For example, when antigen presenting cell express an antigen on MHC class 2, a CD4 cell will aid those cells through the combination of cell to cell interaction, that is CD40 protein and CD40L, and through the cytokine. So in this diagram we can see this is the antigen presenting cell with MHC class 2 protein peptide, and this is the CD4 T cell. The CD4 T cell, it has got a receptor on it which can bind with the peptide. It has got CD4 molecule and it forms a complex, and it is responsible for the release of cytokines, and ultimately these cytokines, they enhances the B cells to produce more antibodies. They enhances the macrophages to go for more phagocytic activities. They enhances the killer T cells to show the cytotoxicity. So this is a function of CD4 or helper T cell. Next terminology is MAC, that is membrane attacking complex. Membrane attacking complex of complement system refers to the association of complement proteins, that is C5b, C6, C7, C8 and C9, to form potentially cytolytic complex. When C5 is activated by either the classical or alternate pathway, the resulting C5b molecule contains binding sites for the next components in the pathway. Electron microscopic studies suggest that C5b and poly C9 complex forms a hollow cylinder. This cylinder is formed by the polymerized C9 molecules which number between 12 to 18 in each complex. This forms a pore in the cell membrane similar to the perforin pore of cytotoxic T cells and NK cells. The pore renders the cell permeable to small ions but not to the proteins, and is therefore thought to cause cell death by osmotic effect. So in this diagram we can see the formation of membrane attacking complex with the help of poly9. Next terminology is hapten. Hapten is a relatively small molecule that elicits an immune response only when attacked to large carrier such as a protein. The carrier may be one that also does not elicit the immune response by itself. Once the body has generated antibodies to hapten carrier adduct, the small molecule hapten may also be able to bind to the antibody, but it has usually not initiate an immune response. In the early part of 20th century, immunologist Karl Landsteiner exploited the antigenic quality of synthetic haptens to study the highly specific way in which antibodies bind to the antigen. The first researched hapten was aniline and its carboxyl derivatives ortho para-amino benzoic acid. So in the case of haptens, it binds with the carrier molecule, forms hapten carrier complex, which leads to induce the immune response. Haptens themselves cannot. Next term is adjuvants. Adjuvants improve poorly immunogenic vaccines. Adjuvants are substances used in combination with specific antigens that produce a more robust immune response than the antigen alone, thus enhancing a wide range of materials. Adjuvant affect the immune response in various ways: to increase the immunogenicity of weak antigen, to enhance speed and duration of immune response, to stimulate and modulate humoral responses including the antibody isotype, to stimulate cell-mediated immunity, to improve induction of mucosal immunity, enhance immune response in immunologically immature patients, particularly infants, to decrease the dose of antigen required, reducing the cost and eliminating inconvenient requirement for the booster shots. So many molecules have been considered for use as adjuvant, like minor mineral compounds like alum. First use. Then is water in oil, oil in water emulsions. They are FREUND'S adjuvant. Natural and synthetic toxins derived from bacteria, that is cholera toxin, CT, lymphotoxin. Based on their mechanism of action, adjuvants have been categorized into two broad groups. Number one, particulate vaccine delivery system that target antigen to antigen presenting cells. Then second is immunostimulatory adjuvants that directly activate such cells through specific receptors like toll-like receptor, resulting in inflammatory response which amplify the innate immune response. So the ultimate aim is to activate the immune system to respond more rapidly to infection and for adaptive immune response to become more specific. So these are the various terms which are used in the immunological study. I hope the terms they are clear to you. Thank you.